Conquest, and we are going to be going over protein synthesis today. And before we start, I want to make sure that you guys know the structure of DNA. Hopefully, last week you kind of understood this and know some of the base pair rules. So let's start by looking at the PowerPoint and just doing a quick review. All right, so first off, before we get started, some quick review or reminders about DNA. So all living things have DNA, whether you're a human, an animal, a plant, we all have DNA. It's found in the nucleus, and it cannot leave the nucleus, so it's stuck inside the nucleus, and it controls all of your genetic information. So your DNA is specific to you. All right, so DNA was discovered about 100 years ago by Frederick Griffin, and the two scientists, Watson and Crick, they're the ones who discovered the structure as a double helix. So basically there's two strands and it looks like a spiral staircase. Each part of your DNA is made up of nucleic acids. Right? So the DNA strands are not identical, rather they complement each other. So here's where the base pairs come in. Okay, so DNA is composed of nucleotides. I said nucleic acids. I meant nucleotides. And each nucleotide contains a sugar, a phosphate, and the nitrogen base. These here in the center are your bases. They're like the steps of the stairs. And they pair together accordingly. So here's my nucleotide. Here's my base. The names of these bases you don't have to remember. However, you should at least know the first letter. So we have guanine, cytosine, adenine, thymine. By now, you should know A pairs with T and C pairs with G. So if you remember apples in a tree, cars in a garage, you won't forget that. All right, so real quick, let's do a practice. So I'm going to have you guys fill this in and submit your question, your answer to this question. So CTCG. Good. All right, lastly, we want to remember that DNA is the universal code, which means universally every living thing has it. In addition to that, we all have those same four bases, A, T, C, and G. The thing that makes us all different or unique is the order of these bases. Think if you had a combination lock and it was 4 numbers, and I didn't give you the correct sequence of numbers, you would have to try millions of times to get into that lock. Same thing with our DNA. Now, DNA has to replicate, means it makes a copy of itself, and this happens in interphase, specifically the S phase, if you remember from last time. And when it makes a copy of itself, it does the base pair rules, so the A, the T, C, and G. Now, answer this question. I just gave you the answer. What phase does DNA replication take place? Okay, let's practice one more time. Take a minute and fill in the corresponding letters that pair with the DNA sequence. All right, now, that's all review. When DNA replicates, you probably haven't gone into this yet, but there's an enzyme called helicase that unzips it. So it separates the two strands of DNA. Then another enzyme helps add the free nucleotides to the correct pair. In the end, you get 2 identical strands of the original strand for DNA. Let's see what that looks like. So an enzyme called helicase unzips the DNA. See how it now goes to 2 sides? You have free-floating nucleotides, so they will pair up to the corresponding base pair. And our end result, we have 2 strands of DNA. So that is DNA replication. All right, today we're going to focus on why DNA has to turn into RNA, and why it's so important. So let me take this off. And I'm going to share my dot cam with you. If you have paper, I suggest drawing this out with me. Okay? So let's see here. We're going to go very basic. All right, to start off, we're going to draw a cell. Nice and big. All right, so this particular cell is a eukaryote, which means it has a nucleus. So if this is my nucleus inside my cell, I'm going to have to know that DNA is found in the nucleus. All right. Now, DNA cannot leave the nucleus. So let's write that off here. Cannot leave nucleus. Why is because DNA itself, it's too big. Okay, so physically, it's like trying to fit an elephant through a door to your classroom. It won't fit. So we should know from the previous couple units the organelle that makes proteins. All right. Anyone want to take a guess? It is a ribosome. So ribosomes are responsible for making proteins. Okay. So I'm going to draw a ribosome. Not drawn scale, but you get the idea. Here's the nucleus. All right, so DNA is the most important thing inside your cell. It has all the instructions. It has the instructions necessary for the ribosome to know what kind of protein to make. But if DNA cannot leave the nucleus, well, how does the ribosome get those instructions? Basically, what happens is DNA has to change itself into RNA. RNA is a single strand, unlike DNA. So if here's DNA, okay, I have a double helix with my base pairs. Well, RNA is a single strand, so it's much smaller, and it can actually fit through the pores of the nuclear envelope. So DNA has to turn into RNA. Once that happens, DNA basically gives RNA the message to send to the ribosome to tell it how to make the protein. So we often call this messenger RNA or mRNA. Once mRNA is formed, it takes the message to the ribosome, and the ribosome starts to work on the protein. Now, if you recall, amino acids are what makes proteins. They are the building blocks. So if I have several different amino acids paired up in a row, kind of like a beaded necklace, this whole thing is my protein. These individual units, the circles, are the amino acids. Okay. Now, what we need to learn is how does messenger RNA tell the ribosome what amino acids are needed to build a specific protein? All right, so let's look at something else real quick. Actually, let's look at Remember that DNA has the 4 bases. We have adenine, thymine, cytosine, and guanine. All right, A, T, C, G. So A pairs with T, C pairs to G. This is specifically DNA. RNA is similar, however, there is no thymine. It has adenine. Instead of thymine, it has uracil. Okay, so now cytosine and guanine stay the same. So A pairs with U, C now pairs with G. So let's look at an example. All right, you guys should have this paper. It's giving me my DNA sequence, and it wants me to change it to messenger RNA. So A, normally in DNA would turn to T, but we're doing RNA, so now A has to turn to U for uracil. G and C don't change, so G still becomes C, C becomes G. C becomes G. A again, here we are, becomes uracil. G, C. T still turns to A because we do have adenine in RNA. So I'm going to go ahead and finish this up. T turns to A, G, A, U, U, G, G. So there is my strand of messenger RNA. Notice how your paper has these letters kind of grouped in threes. There's one, there's one, there's one, there's one, and then the last one. These are paired this way because they are called codons. So what I'm going to do is I'm going to look at the first strand here of DNA. A, G, C, C, A, G. I'm just going to do the first three, T, A, T. Okay, so this is DNA. If I change it to RNA like I did down here, UCG, GUC, AUA. So here is my RNA. Pull this down. All right, every 3 letters of your RNA is called a codon. So make sure you know that word, codon, C-O-D-O-N. Basically, it's a code for a name of an amino acid. What are you talking about? All right. Look at your codon chart. So this chart right here is going to help us uncode the name of the amino acid. Kind of cool. It looks like a star or something you find on a pirate ship. My first letters, again, U, C, G. So I'm going to take these 3 letters and use my decoder, UCG, to find the name of the amino acid. So how it works is you start in the middle with the largest letter. So my first letter is U. So I'm going to start here with U. Okay, my next letter is C, so I'm going to move outward to the next smallest letter. So here's the C. And then G is the last letter. So here. So because it goes straight like this, the amino acid's name that I'm looking for is serine. So what you would do is you would write the name of your amino acid right here. Okay, let's practice the next one and then I'll let you guys do the rest. So we have G-U-C. All right, I'm starting over here now. So here's my G, here's my U, here's my C. So this amino acid is called valine. So you would write valine right there. Okay, I'm gonna let you guys do the next 3. All right. So coming back to this, let's just recap what we've learned. Your cell has the nucleus where DNA is found. DNA, excuse me, has the instructions for the ribosome to make the proteins. However, the DNA cannot leave the nucleus because remember, too big. So in order to get the instructions to the ribosome, it has to change into messenger RNA. All right, so we practiced doing that. The only difference is there's no thymine. Instead, we have uracil. All right, so as I changed DNA into RNA, I now have each codon I need in order to uncode the name of the amino acids. So we discovered this one was serine, right? So that means that if I'm using this protein, that the first amino acid is going to be serine. Does that make sense? GUC, if you remember, was valine. So this codon is my next amino acid, which would be valine. Right? Sometimes, and you'll find this on your practice, the last amino acid would be called a stop codon. And basically, that's just telling the protein that it's done being made, stop, process is over. All right, so if you see that, don't get thrown off. All right, so your goal for today is to finish filling this paper out, decode all of the amino acids, okay? If you get stuck, ask a friend, or you can come see me in room 1312. All right, let's see. So that's the end of this lesson, protein synthesis, which means making proteins. Remember, proteins are made by the ribosome, and DNA has the instructions to make the proteins. But DNA has to give those instructions to RNA because RNA is the only one that can leave the nucleus and take it to the ribosome. All right, I hope you guys understand this. Tomorrow we'll go a little bit deeper, but pretty much this is the bulk of protein synthesis. So you can do that, you're on the right track.